Protein factories. Yes, ribosomes. You know about it, right? Ribosomes, they're the protein factories discovered by George Palade, 1953, using electron microscope. Okay, once more, the story that we have talked about, just keep that in your mind, because once we cover few organelles, then you will be able to relate to it. Okay, slowly. So discovered by George Palade, 1953, using electron microscope, and that's the reason it's also known as the Palade particles. You know, it's made up of RNA and proteins. Fine. Smallest cell organelle. Ribosomes, they are the smallest cell organelles. They are present. All living cells have ribosomes except, please note this down, except the mammalian RBCs. All living cells have ribosomes except the mammalian RBCs. And they are, again, you have to note this down, non-membrane bound organelles. Before this, we have studied about the membrane bound organelles, and that too double membrane. Here, these are non-membrane bound organelles. Fine. Note these 2 points down. Come on. What is the size? About 50 nanometers by 20 nanometers in size. So you understand, quite small. Smallest cell organelle. Okay. So these are, these are dense structures. They are not fluid filled. They're not fluid filled structures. These are quite dense structures made up of the RNA and the proteins. Correct? Let me show you. Do they have some subunits? Yes. We have talked about it. They do have subunits. Let's see. There are 2 subunits, as you can see on the screen. Yes, 2 subunits. And what are they? This is the larger subunit. This is a smaller subunit. 2 subunits. You know what? These 2 subunits together, they function as a proper functional ribosome and helps in the protein synthesis. Yes. So both these subunits are essential. Correct? Take a look at it. Let me show you properly. Let's take a proper 3D view. Okay? Take some time and try to see the structure. So this is the ribosome. It's the whole structure. This is the larger subunit. Correct? And this is the smaller subunit. And together, when they have combined together, assembled together, then it is a functional structure. Got it? Which is capable of protein synthesis. Now, types of ribosome. Do we have types? Yes, definitely we have types. So these are the 2 major types of ribosomes, 70S and 80S ribosomes. Where are they present? We have studied about it. Prokaryotes, they have 70S ribosomes, and eukaryotic cells, they have 80S ribosomes. That's the difference. Prokaryotes have 70S ribosomes, eukaryotes have 80S ribosomes. Now the question is, do you recall we have studied about some eukaryotic cells and then related to the 70S ribosomes also? Can any one of you tell me? And let me see who is following the sessions properly. Some eukaryotic cells, cell organelles, the next hint. You've talked about some organelles, and there we have talked about the 70S type of ribosomes. Can you tell me that quickly? Come on. Yes? Okay, I'll not declare the answer right now because it's coming up. Wait for it. So now, okay, what is this S? I've been talking about this in many sessions of ours. S, 70S, 80S. What is this S? Okay, let's take the opportunity of this session and try to understand what's this S. Well, S, it stands for the Svedberg unit. It's basically the sedimentation coefficient. You know what is sedimentation, right? Sedimentation? That's a simple term. Sedimentation. A measure of how quickly particles sediment in an ultracentrifuge. Well, does any one of you know what is a centrifuge? Look here, what's happening. You see some tube-like structures. There are specific names for these. Don't have to worry about this right now. Some tube-like structures, they are placed, and see, initially they are placed straight. And when it starts rotating, see, it's moving, right? What's happening? When spinning, they change their angles. They're changing their angles, right? They are moving outwards. Can any one of you tell me why? Why? Or rather, can you give me a very simple example which can actually portray this particular mechanism? And in fact, that is the mechanism we have used in developing centrifuge, this machine. And this technique is known as centrifugation. Okay, centrifuge. I'll give you an example. Just think about, you try it out yourself. In physics, I'm sure you have studied about this. Take a thin rope, okay, thin string also works, and then tie a weight. It can be a pebble, it can be anything, just a weight, right? And then what you'll do, you'll take that and keep spinning. Spin it, okay? Spin it, and then at one point of time, do one thing, release it. If you do that, what will happen? What will happen exactly? You have to give me the answer. I'm spinning, okay, noted. I'm spinning, and I release it at here, this point. So let's imagine let me take a pebble, okay? Pebble is, the position is here, it's moving, and when it has come here, I've released it. Which side it will go? Will it rotate? Will it take a round again? Yes. So will it spin? Rather, I would use the word spin, because rotation, revolution, there are different forms based on the axis. So let's not dive there. Spinning, right? So if I leave at this position, which side will it go? Yes. Recall about the centrifugal force. Centrifugal force, because of which, if I release at this position, it will try to move this way, tangential. Correct? Yes or no? Yes. So the same process or same principle is used over here. So when we are spinning at a high speed, what happens? Based on the density of the particles, they separate down. Okay? They separate in a fluid medium. They will separate. Correct? So basically, with this technique, it's an indirect measure of the density and the mass. Density and the size. Density and size. Indirect measure. So what I can say is, the larger and the denser, if we rub it here, yes. So larger and denser the particle, greater will be its S value. Right? Smaller and lighter the particle, lesser will be the S value. Now tell me, if there are two particles here in this medium, one has, let's imagine, yes, 70S and 80S. Fine. Which is the particle which is larger or denser? They will sediment more towards the bottom, and particles which are lighter, they will be towards the up. Okay, upper side, upper layers. So that's simple because of this spinning movement, centrifugal force, density and size. So these are the factors. Got it? So yes, we have utilized this technique, this principle, and based on that, the types of ribosomes, 70S and 80S. As you remember now, the Svedberg unit, subunits synthesized in the cytoplasm. Okay, 70S subunit synthesized in the cytoplasm, and 80S subunit synthesized in the nucleolus. So the sites of synthesis of these two types of ribosomes. Prokaryotic cells, yes, they have 70S, and if I talk about the eukaryotic cells, they have 80S. Do you take a look over here? If I am talking about 70S, 50S and 30S. Okay, but 30 and 50 is 80, but I am referring to it as 70S. 60 and 40 is 100. I am referring to as 80S. Well, the basic logic is, or the basic principle behind is this, it's not additive. It's basically, whenever these two molecules are present together, what is this S? S is basically this sedimentation coefficient, that measure. Okay, it's not additive. Don't misunderstand this. It's never additive. So 60 plus 40 should have been 100S. But it's wrong. It's not 100S. It's 80S, and that's again calculated based on the sedimentation coefficient. That's it. Okay? Well, so, only the subunits are synthesized in the nucleolus, and then they're transported to the cytoplasm through the nuclear pore. Remember about the nuclear pore? And there they will assemble and then act as a functional protein synthesizing unit. Got it? Okay. Magnesium ions. These are essential for the binding of the ribosomal subunits. Right? Okay. Well, did you know about this? Organelle within an organelle. Organelle within an organelle. Yes, mitochondria has 55S type of ribosome. If I talk about chloroplast, yes, I asked you this question, right? They have 70S type of ribosome. Now, another new thing you are going to learn right now is mitochondria. Remember, we have studied about the 70S ribosome, presence of 70S ribosomes in the mitochondria as well as chloroplast also. But here I am talking about the 55S. You know, this is a specific characteristic feature of the mammalian ribosomes. Correct? If you talk about the eukaryotes, the mitochondria of the eukaryotes, they have 70S type of ribosome. But if I talk about the mitochondria of the mammals, they have 55S type of ribosomes. So please note it down. Note it down. This is important with respect to your NEET examination. Chloroplasts, we have already studied, they have 70S type of ribosomes. Fine. So you understand, organelle within an organelle. Correct? Mitochondria, it's an organelle. Inside that they have the ribosomes, another organelle. So organelle within an organelle. Same is the case for chloroplast. Okay, where else are ribosomes found? Let's see. What is this? Rough endoplasmic reticulum. Endoplasmic reticulum. Yes, rough endoplasmic reticulum. The ribosomes are again found over there. Are found embedded to the rough endoplasmic reticulum. In addition to that, functions of ribosome, if I talk about protein synthesis. Yes, polysomes. We studied about this. Polyribosomes, group of ribosomes bound to the mRNA. We'll study about what is mRNA in your higher standards, in 12th standard, right? But just remember, this is a structure which has the codes which will result in the protein synthesis. Fine? Yes, and who helps that? Ribosomes. Synthesize proteins. Major function of the ribosomes. Important, very important function. See, direction of the movement of ribosomes over here. And why called polysomes? Because multiple ribosomes are sitting on a particular single mRNA and it is translating. Okay, it is just synthesizing the proteins. That's it. Fine. Okay. Now, see, growing polypeptide chain. These are the growing polypeptide chains. Great. This is a microscopic view of polysomes under the microscope. Look how they look under a microscope. It's so different, right? Okay. Now, from cytoplasm to the endoplasmic reticulum. Wow. What happens here? Ribosomes bound to the rough endoplasmic reticulum produce proteins. Ribosomes which are bound to the rough endoplasmic reticulum, and that's the reason it's called rough. I will be talking about it in detail. But just remember, endoplasmic reticulum with which the ribosomes are associated, they are known as the rough endoplasmic reticulum. They produce the proteins. So proteins produced by a transfer to the rough endoplasmic reticulum lumen. This is very important. Let's see. Look at the picture. Do you see the ribosomes? Correct. mRNA is there. Now, see the peptides. Peptides, after they are synthesized, they are transferred into the lumen, the inner part of the endoplasmic reticulum. That's a lumen. Correct?